Good morning, students. Hope you all are fine and safe at home. In the last video, we discussed about dihybrid cross and law of independent assortment. Now we will discuss about special cross. Basically, we are going to talk about back cross. It is further of two types, out cross and test cross. Back cross is actually a standard procedure in breeding programs. The original cross for obtaining hybrid is made to introduce one or more useful traits, like it may be related to useful trait may be related to the disease resistance, pest resistance, high protein content, high vitamin content. So to obtain these good characters, back cross are made to reintroduce all the good characters of original variety. ओरिजिनल वैरायटी के गुड करेक्टर्स को लेने के लिए क्या करेंगे हम लोग बैक क्रॉस करवाएंगे. Now what is out cross? When F1 individual is crossed with dominant parent, it is termed as out cross. जब हम F1 individual को dominant parent से cross कराएंगे तो इसे हम बोलेंगे out cross. And the generation obtained from this cross all will possess the dominant character. जो generation obtained होंगी all will possess the dominant character. You can see with the help of this cross. As you know, in monohybrid cross, we obtain F1 generation as all told, capital T, small t we obtain, right? That hybrid tall plants. Now what we have to do, we have to cross F1 progeny with the dominant parent. Dominant parent was pure tall or homozygous tall parent. Now we will receive here two gametes, capital T, small t, and here also capital T, capital T. Now what will happen, gametes capital T, small t, and here the dominant parent capital T, capital T. So what we obtain here, capital T, here also capital T, here capital T, small t, and here capital T, small t. So what we observe that progeny or the generation obtained from this cross possesses dominant character, as all the progeny that was obtained was tall, right? So this is what I have already told you, that when F1 individual is crossed with dominant parent, then it is termed as outcross, and the generation obtained from this cross possesses dominant character. So any analysis cannot be possible in F1 generation because we got all the progenies that were dominant, that were tall, that were possessing the dominant character. Test cross. When F1 progeny is crossed with a recessive parent, then it is called as test cross. When F1 progeny ko we will cross with recessive parent, then it is termed as test cross. Now what is the usefulness or the advantage of this cross? It will help us to find out the genotype of an organism. So what we actually do here, in this cross, the organism with unknown dominant genotype is crossed with recessive parent. We will find out the genotype of an organism with the help of test cross. How? Firstly, we will discuss about a monohybrid test cross. The progeny obtained from monohybrid test cross are in equal proportion, means 50% is dominant phenotype and 50% is recessive phenotype. We can show it like this. In F1 progeny, what the hybrid is shown as capital T and small t, right? This is F1 hybrid. It is crossed with the recessive trait or the recessive parent, that is small t, right? We'll obtain here capital T, small t, and from here small t, small. I will show you this cross with the help of Punnett square, showing here the hybrid. Now actually they are the parents. Here I will show the recessive parent. What you will obtain, capital T, small t, capital T, small t, small t, small t, and here small t, small t. So what you observe, that in the monohybrid test cross ratio you obtained 1:1. How? Because 50% of the parent, 50% of the progeny is tall, denoting as capital T, small t, and 50% of the progeny is small t, that is dwarf. So we obtained 1:1 as phenotype as well as genotype, 1:1. Now we will discuss dihybrid test cross. Okay, the progeny obtained from dihybrid test cross are 4 types and each of them is 25%. In F1 hybrid of a dihybrid cross, assume we have taken 2 character of tallness, that is the height of plant, and the seed shape, round and wrinkle. The hybrid will be shown like this. We will cross it with recessive parent. Recessive parent was dwarf with wrinkle seeds. Okay. Now I am showing here the Punnett square. So gametes obtained from this parent will be small t and small r, right? Whereas gamete obtained from this parent will be capital R, capital T, small r, capital R, small t, and small t, small r. So here you will obtain that capital T, small t, capital R, small r. Here capital T, small r, right? Here small t, capital R, small r, small t, small r, right? We will obtain these characters. Here capital T, small t, capital R, small r, capital T, small r, small r, small t, capital R, small r, and small t, small r. So the ratio of dihybrid test cross will be 1:1:1:1, right? And this will be the phenotypic ratio. And it will show tall round, it will show tall wrinkle, it will show dwarf round, and it will show dwarf wrinkle. Okay, so ratio of dihybrid test cross will be 1:1:1:1. So what conclusion we can draw from this monohybrid test cross ratio as well as dihybrid cross ratio? Actually, this conclusion can be drawn that in test cross, phenotype and genotype, we observe that phenotype and genotype both are same, right? And second inference was this: the test cross actually helps us to find out the genotype of dominant individual. Now how this will be able to determine the genotype of an individual, dominant unknown individual, we will discuss this with the help of example, and this example is mentioned in your NCERT textbook. Here also a cross is shown, a monohybrid cross is shown between violet color flower, which is denoted as capital W, and a white color flower, which is denoted as a small w, right? And the F1 hybrid was violet color. F1 hybrid जो obtained हुआ वो violet color flower था. तो टेस्ट क्रॉस में हमने किया क्या, हम टेस्ट क्रॉस की डेफिनेशन आपको मैंने बताई थी. It's a special type of back cross which is made between individual with the dominant trait and its recessive parent, in order to know whether the individual is homozygous or heterozygous. हमें ये पता लगाना है कि ये जो डोमिनेंट फिनोटाइप है, ये जो डोमिनेंट इंडिविजुअल है, इसका जो जीनोटाइप है वो क्या है. जीनोटाइप हमें नहीं पता, आइदर ये होमोजाइगस है या फिर ये हेट्रोजाइगस है, हमें ये पता लगाना है. ठीक है, ये हम टेस्ट क्रॉस से पता लगाएंगे. तो यहाँ पे हमने क्या किया है, इधर वी हैव एज्यूम्ड दैट इट इज़ होमोजाइगस. सो द gametes release will be capital W, and gametes released from this recessive parent will be small w. So what you observe in this cross, that all the flowers are violet, right? all the F1 progenies are of violet color. So what you conclude from this, that the unknown flower is homozygous dominant. जो unknown flower था वो क्या है, वो homozygous dominant है. Whereas अगर आप heterozygous ले रहे हो तो what you actually observe here, the gametes release will be capital W and small w, and gametes release from this homozygous recessive parent will be small w. So you observe that half of the flower, that is 50% of the flowers were violet and half of the flowers were white, and 50% were white. That is ratio obtained is 1:1. From what you conclude from this result, that unknown flower is heterozygous. जो unknown flower है इसका genotype क्या है, heterozygous. So this is all about test cross. From this cross you can identify or you can determine the genotype of an organism, and in this cross the organism with unknown dominant genotype, this is unknown dominant genotype, is crossed with the recessive parent. We have crossed it with the recessive parent. These were the recessive parents actually. So this is all about test cross. There is another type of special cross that is termed as reciprocal cross. Now what exactly is reciprocal cross? When two parents are used in two experiments in such a way that in one experiment, in the first experiment, A is used as female parent, you using A as a female parent and B as male parent. Okay, in the other experiment, say it's the second experiment, A will be used as the male parent, you are using A as male parent and B as female parent. Such type of sets of two experiment is called reciprocal cross. This type of cross in which you reverse the male and female parents, this is termed as reciprocal cross. As you can see here, assume this is the first cross where I have taken capital T crossed with small t. But in this cross, this capital T denotes a female parent, whereas this small t denotes male parent. Okay, now what you will obtain, that all the plants will be tall, right? Because from here we will receive gamete capital T and from here small t. Now assume a second cross where we take capital T crossed with small t, but this time this capital T we have taken the male parent and small t as female parent. So what inference you can draw from here, that here also you will obtain that all the F1 progeny will be tall. Okay, so actually the characters which are controlled by carrier gene are not affected by reciprocal cross. What is mean by carrier gene? Actually, Mendel obtained same result. This cross was performed by Mendel, and Mendel obtained same result after reciprocal cross because Mendelian characters were controlled by carrier gene which is present on autosome. Whereas Morgan, he reported variations or changes during reciprocal cross in sex linkage as well as cytoplasmic inheritance. We will discuss about Morgan experiment in detail. So this is all about reciprocal cross. Next we are going to discuss about chromosomal theory of inheritance. Actually, Mendel's result could not be accepted immediately by the scientific world, as we have already discussed earlier in the previous videos, because of the findings that traits are controlled by stable, non-blending and discrete units called gene or factor. His mathematical derivation and explanation of biological phenomena were also not accepted by many scientists of that time. However, discovery of chromosomes, their replication and separation during mitosis and movement during meiosis indicate some parallelism between them and hereditary factor. It is called as chromosomal theory of inheritance. This theory was proposed by Sutton and Boveri, and there are some of the features of this theory. We are going to study it right now. The genetic material of eukaryotes is distributed among multiple chromosomes. ये थ्योरी क्या बोलती है कि जो जेनेटिक मटेरियल है यूकैरियोट्स का, इट्स डिस्ट्रीब्यूटेड अमंग मल्टीपल क्रोमोसोम्स पे जेनेटिक मटेरियल क्या है, डिस्ट्रीब्यूटेड है. इन 1902 Sutton and Boveri independently recognized that transmission of chromosomes from one generation to the next parallels the pattern of transmission of Mendelian factors from one generation to the next. इन्होंने क्या बताया कि जैसे ट्रांसमिशन हो रहा है क्रोमोसोम्स का एक जनरेशन से दूसरी जनरेशन में, सिमिलरली जो मेंडल के फैक्टर्स थे या जो जीन्स थे वो भी एक जनरेशन से दूसरे जनरेशन में ट्रांसफ़र हो रहे थे, ट्रांसमिटिंग द इंफॉर्मेशन फ्रॉम वन जनरेशन टू नेक्स्ट जनरेशन. They proposed the chromosome theory of inheritance, which states that chromosomes are the carrier of genes. Chromosomes क्या है, ये carrier है genes के, that is the genes are located in chromosome. इन्होंने बोला कि जैसे कि chromosome है और इसके ऊपर क्या present है, genes present है, which will carry the genetic material or the genetic information from one generation to the next generation. Now, some of the evidences were considered to explain the chromosomal theory of inheritance. That's the number of chromosome is constant from cell to cell within an organism, from organism to organism within any one species, and from generation to generation within that species. क्या बोला इन्होंने कि जो क्रोमोसोम नंबर है वो कांस्टेंट रहेगा फ्रॉम सेल टू सेल विद इन एन ऑर्गेनिज्म, फ्रॉम ऑर्गेनिज्म टू ऑर्गेनिज्म विद इन एनी वन स्पीसीज. कोई स्पीसीज कंसिडर कर ली, उसमें जो सभी ऑर्गेनिज्म है उनमें जो क्रोमोसोम नंबर है वो क्या होगा, कांस्टेंट रहेगा. फ्रॉम जनरेशन टू जनरेशन विद इन दैट स्पीसीज, कोई पर्टिकुलर स्पीसीज कंसिडर की उसमें नेक्स्ट जनरेशन मतलब वन जनरेशन टू नेक्स्ट जनरेशन क्या हो रहा है, क्रोमोसोम नंबर जो है वो कांस्टेंट रहेगा. Second point, what they consider was eukaryotes have two copies of each type of chromosome in their nuclei. The chromosome complement is said to be diploid. उनके क्रोमोसोम को हम क्या बोलेंगे, डिप्लॉयड या 2n. Paired homologous chromosome separate during gamete formation, ये हमने पढ़ा था that कि जो homologous chromosome का पेयर है वो क्या होगा, गैमेट फॉर्मेशन के टाइम पे सेपरेट होगा. Separate during gamete formation. Thus, gamete contain half as many chromosomes as somatic cells of same organism. That is, each gamete has only one set of chromosome, and it is said to be haploid. Diploid individuals are produced by the fusion of two gamete nuclei, one from male parent and another from female parents. Let's discuss in short that what they want to say. Actually, they want to say that one of the copy of chromosome or one of the haploid set is received from the male parent and one haploid set is received from female parent. Although after the fusion of gametes we have a diploid set, that is 2n, yani organism will be diploid. Haploid set n and haploid set n is received from both the parents, and after the fertilization or the fusion we received the 2n. After fertilization, diploid individual is formed, or you can say that individual form will have a diploid set of chromosome. Therefore, the behavior of Mendel's factor or genes during the production of gamete in bees precisely parallels the behavior of chromosome at meiosis. You must have read about mitosis and meiosis. Mitosis is termed as equational division, whereas meiosis is termed as reductional division. You must have read about it in 11th class. But they concluded that the behavior of chromosome and the behavior of genes or factors as said by Mendel during the production of gametes was similar. They said genes are in pairs, so are chromosomes. Gene pair में थे, chromosome भी pair में है. The alleles of gene segregate equally into gametes. जो alleles होते हैं एक gene के वो क्या करेंगे, segregate होते हैं, separate होते हैं equally into gametes. So do the members of a pair of homologous chromosome. उन्होंने genes की बात की थी जो क्या हो रहे थे, gamete formation के टाइम पे एक दूसरे से separate हो रहे थे, like this. They were separating, right? And what they said, like this is a pair of chromosome. During gamete formation they will segregate from each other or separate from each other. Different genes act independently, so do different chromosome pairs. अब जो different genes है, मैंने बोला था कि वो independent है, they are free to form any combination, so do the different chromosomal pairs. Next point, except during gamete formation, genes must be accurately reproduced and transmitted to the daughter cells in such a way that each cell contains a set of identical to its parent. हर एक सेल के अंदर जो एक सेट आए वो आइडेंटिकल हो अपने पेरेंट्स के. सो क्रोमोसोम्स टू आर वेरी केयरफुली रेप्लिकेटेड. क्रोमोसोम्स का भी जो रेप्लिकेशन होता है वो भी बहुत केयरफुली होता है. एंड सेपरेटेड ड्यूरिंग सेल डिवीजन सो दैट नॉर्मली ऑल डिसेंडेंट्स ऑफ अ सेल कंटेन एक्यूरेट कॉपीज ऑफ क्रोमोसोम. ताकि जो जितना भी के डिसेंडेंट्स है सेल के उनमें क्या हो, एक्यूरेट कॉपीज आए उनके क्रोमोसोम की. नेक्स्ट पॉइंट, visible changes in chromosome structure can often be correlated with genetic changes. Visible changes can be seen and you can correlate it with the genetic changes. Example, the arms of two homologous chromosomes sometimes exchange part during meiosis. This also you have read like this. Consider this. This is the two arms of a chromosome. Okay. So these arms of two homologous chromosomes sometimes exchange part. Here at this site, some part will be exchanged during the process of meiosis, and the visible feature of this exchange is a joining between arms of two chromosomes at a point known as chiasma. You must have read about chiasma. It is formed like this. See here. This is one chromosome. This is another chromosome. So X-shaped structure is formed at this site. The site of exchange of genetic material is termed as a chiasma. The chiasma was found to correlate with interchange of allelic genes between homologous chromosomes. So these homologous chromosomes will exchange the genetic information or the genetic material at this site. This is termed as chiasma. These are X-shaped structure. This parallel behavior of genes and chromosomes suggests that genes are located on chromosome. तो ये जो अभी हमने behavior study किया genes और chromosome का, इनको relate किया, तो हम इनसे ये conclusion निकाल सकते हैं that the genes are located on chromosome. So this is how you can see the correlation between chromosomal behavior and Mendelian factor in segregation and independent assortment. These are the two alleles which are present on the same locus of the homologous chromosome. This is denoting yellow and it is denoting green. This is round and wrinkle. Homologous chromosome. Okay. Independent assortment. Both of the alleles will segregate and they are independent to form any combination. What are some of the similarities which we can draw between genes and chromosome? As we have discussed just now that genes as well as chromosomes are transferred from generation to the another generation without any change. हमने स्टडी किया कि जो genes और chromosome दोनों ही ट्रांसफ़र होते हैं एक जनरेशन से दूसरी जनरेशन में without any change. And in diploid cell chromosome occur in homologous pair. diploid cell में जो chromosome है वो occur करते हैं homologous pair के अंदर. And gene also occur in allelic pair. One member of pair is obtained from paternal parent, that is one member of pair is obtained from your father and other from maternal parent, that is from your mother. And prior to cell division each chromosome as well as allele of genes get replicated. cell division से पहले क्या होता है, हर एक जो chromosome है या जो alleles है जिनकी वो replicate होती है. replicate means forming its copy. During mitosis the replicated chromosome as well as the replicate allele splits. Then mitosis के टाइम पे क्या होता है कि जो replicated chromosome है as well as जो replicate allele है वो split होगी. The split function as a daughter which pass into two daughter cells. Like this is an, assume it is an allele, it will split and it will pass equally in the two daughter cell. The split functions as daughter which pass into two daughter cells. The method of replication and distribution maintain exact similarity in genetic composition of the cells of multicellular organism. ये जो मेथड है रेप्लिकेशन का और जो ये डिस्ट्रीब्यूशन है ये मेंटेन क्या करेगा कि एग्जैक्ट सिमिलैरिटी रहे, जो जेनेटिक कंपोजिशन है सेल्स का मल्टीसेलुलर ऑर्गेनिज्म के अंदर वो सेम रहे, उसके अंदर सिमिलैरिटी रहे. एंड बोथ सेग्रीगेट ड्यूरिंग गैमिट फॉर्मेशन. और क्या होगा, गैमिट फॉर्मेशन के टाइम पे वो सेग्रीगेट हो जाएंगे और मियोसिस के टाइम पे, सो दैट अ गैमिट कम्स टू हैव ओनली वन क्रोमोसोम एंड वन अलील ऑफ ईच पेयर. ताकि हर एक गैमिट के अंदर क्या हो, या तो एक क्रोमोसोम आए या अलील आए. यानी अगर हम क्रोमोसोम के बारे में बात करेंगे तो एक क्रोमोसोम आए, और अगर हम जीन को लेके चल रहे हैं, फैक्टर्स को लेके चल रहे हैं, तो एक अलील आए. एंड नेक्स्ट द फ्यूजन ऑफ टू हैप्लॉइड गैमिट्स. और जब ये हैप्लॉइड गैमिट्स जो होंगे एज एन एंड एन, फ्यूजन ऑफ दीज टू हैप्लॉइड गैमिट्स रीस्टोर द डिप्लॉइड क्रोमोसोम नंबर. उससे वापस जो डिप्लॉइड क्रोमोसोम नंबर है वो रीस्टोर हो जाएगा. एज वेल एज अलीलिक पेयर इन द ऑस्प्रिंग. यही हमने मेंडल के लॉज में भी पढ़ा था. अलीलिक पेयर भी आफ्टर फ्यूजन और फर्टिलाइजेशन they will form or they will restore the diploid number. Both chromosome as well as alleles follow law of segregation. क्रोमोसोम और एलील्स दोनों ही लॉ ऑफ सेग्रीगेशन और लॉ ऑफ इंडिपेंडेंट असॉर्टमेंट दोनों को ही फॉलो करती हैं. Hence this is all about chromosomal theory of inheritance, and in the next video we will study about linkage and recombination studied by Morgan.